In this video, we're going to be discussing carbon, hydrocarbons, and functional groups. And the learning objectives for today is that by the end of this, I want you to be able to explain the properties of carbon that make it so important for biology. I want you to be able to identify all of the different hydrocarbon isomers. So how can a molecule made up of nothing more than hydrogen carbons, how can it make up so many different types of structures? And finally, I want you to be able to identify the functional groups on molecules and describe what chemical properties these functional groups contribute to that molecule. So let's start with carbon. Carbon is the element of life, and the primary reason is that it has a valence number of 4. So what that means is it's in its outer electron shell. There are 1, 2, 3, 4 electrons. And remember, the outer shells of this particular outer shell wants to have 8 electrons in it. And so one of the most common ways an atom can fill its outer shell is by sharing electrons with other atoms, and those are called covalent bonds. So here what we have is a carbon forming 4 covalent bonds with 4 different hydrogens. There's one hydrogen, another, another, and another. And notice that hydrogen is sharing its electron with carbon. And so now we've got 2 electrons here, 2 electrons here, 2 electrons being shared here, and 2 electrons being shared there. Each pair of electrons is a covalent bond between carbon and a hydrogen. Carbon and a hydrogen, carbon and a hydrogen. What that means is carbons can have up to 4 covalent bonds with 4 different atoms. And that gives carbon the ability to be involved in lots of different structures. So let's compare that to some of the other elements that are extremely common in life: hydrogens, oxygens, and nitrogens. And what we have is the Honc rule here. Honc, H-O-N-C. And what the Honc rule tells us is how many different covalent bonds each one of these elements or these atoms can be involved in. Honc, H, H, is the first letter, and that means it can be involved in 1 covalent bond. Oxygen is the second letter. Because of its valence number, it can be involved in either 2 covalent bonds or 1 double bond. N is the third letter in the word honc, and it can be involved in 3 covalent bonds, or a triple bond, or a double and a single bond. So all kinds of different possibilities there. And now carbon, having a valence number of 4, means that, as we just saw, it can be involved in 4 different covalent bonds, or any of these possibilities: a double and 2 singles, 2 doubles, a triple and a single. And so when you compare carbon to these other elements, you'll see that there's a greater variety of possible covalent bonds to other atoms, and that variety is what makes carbon special, why it's so important for biology. Because take a look at this. If we take a look at hydrocarbons, if we take a look at molecules that are made up of nothing more than hydrogens and carbons, despite the fact we're looking at molecules that only have 2 types of atoms, look at the variety, at the diversity of structures that we can make simply by connecting carbons and hydrogens in different ways. Right? We can have differences in length, so we can have this carbon chain that is maybe only 2 carbons long, or 3 carbons long, or 5 carbons long, and so on and so forth. And each one of those carbons connected to one another via a single bond and then connected to hydrogens. Or we can now start to look at these carbon chains, still linear chains, but now instead of having every single carbon bonded to the other carbon with a single bond, Now we can start putting in there some double bonds. We got a double bond there. We got a double bond there. And this is not meaningless, right? One butene. So if we have this exact molecule here and we have this one here where they're identical except for the location of the double bond, they will have different chemical characteristics. So the change looks subtle, but it's enough to give these two molecules different characteristics. Now, in these two examples here, we just have carbon chains, linear chains, but we can start adding branches, right? So here's a carbon chain, and then we start a new carbon chain here. That's called branching. Again, they're going to have different chemical properties. And finally, we can take that chain and we can connect the two ends together to form rings. And then on top of that, we can take those rings and we can add some double bonds to them. So all of these differences will lead to molecules with different chemical properties, despite the fact that they are all hydrocarbons. They're all molecules that are made up of nothing more than carbon and hydrogen. Now, the one thing they will have all in common, and don't forget about this, are electronegativities. Always remember your electronegativities. Carbon and hydrogen have about equal electronegativity, which means that no matter what structure we're looking at here, even though they will have some different chemical properties, the one thing that they will share is that they are all non-polar. Okay. Now, obviously, life isn't just hydrocarbons. Life has much more complex molecules than that. And that's why we have to start talking about functional groups. Now, these are little sets of atoms. I mean, here we just have one extra atom. Here we have 4 and 5 atoms, and here we have 1, 2, and 3 atoms that are connected to a carbon. And these functional groups have atoms that are different than just a hydrogen, different than just a carbon, and they are going to give that hydrocarbon some new chemical properties. Now, there's a ton of functional groups out there, folks, but these are going to be the 7 that we are going to focus on, and the 7 that I want you to learn. So here's what I'm saying. If we take a hydrocarbon, isobutane, and we lop off these two hydrogens and replace them with a carboxyl group here and an amino group here, we've made a brand new molecule. We now have valine that has very, very different chemical properties than isobutane because we've got these functional groups here instead of hydrogens, and those functional groups are going to give the molecule a very, very different chemical properties. We can take butane here, again, just a hydrocarbon, a carbon chain of 4 carbons long, and we can lop off that last carbon and replace it with a carboxyl group, and now we have a fatty acid. This butyric acid has very different chemical properties than butane. Okay, so let's take a look at these 7 functional groups. We have a hydroxyl group, a carbonyl group, a carboxyl, amino group, sulfhydryl group, phosphate groups, and methyl groups. You will be required to memorize each one of these to be able to identify them on molecules and be able to describe the unique characteristics, chemical characteristics of each one. All right, let's start with hydroxyl groups. And what a hydroxyl group will look like is an oxygen connected to a hydrogen, and then it's going to be that oxygen that's going to be connected to the carbon. Always look, when we're looking at these functional groups, what part of that functional group is attached to the carbon of that hydrocarbon? And so here's what it might look like in a molecule here. So here's our hydrocarbon, and here is our hydroxyl group connected to it. And so molecules that are primarily just hydroxyls groups are called alcohols. And our hydroxyl group here within these molecules will give that molecule some polarity because oxygen is much more electronegative than hydrogen here, but we're not going to lose that hydrogen. Okay, we're not going to lose that hydrogen proton, leaving the electron here with the oxygen. Okay, it's not ionic. Okay, so hydroxyl groups are polar, but most of the time they're not they are non-ionic. They're not going to lose that hydrogen. Okay, next we have carbonyl groups. These are a little bit complex because we have two flavors. We've got a ketone and we have aldehydes. Okay, regardless of whether we're looking at ketones or aldehydes, what a carbonyl is it is a carbon within our hydrocarbon molecule that is double bonded to an oxygen. There's our carbon that's double bonded to an oxygen. Now, if that carbon is in the middle of a chain, okay, meaning it's connected to two other carbons like we see here, then this is called a ketone. Okay, so a ketone is a type of carbonyl, and it's the type of carbonyl where it exists in the middle of a carbon chain. Look at this one over here. Same thing, we have a carbon that's double bonded to an oxygen, but notice that carbon is at the end of a carbon chain. Okay, that carbon here is only connected to one other. It's not connected to another one because it's at the end of the chain. And we call those type of carbonyls, we call them aldehydes. Okay, so ketones are when the carbonyl is in the middle of a carbon chain. Aldehydes are when they are at the end of a chain. I remember getting this confused like crazy back in the day, so please make sure that you can differentiate those two. Now, like hydroxyl groups, carbonyl groups are polar. Right? Oxygen is going to be carrying a partial negative charge because it's more electronegative than the carbon, but it is not ionic. Next, a sulfhydryl group. Now, a sulfhydryl group is very similar to a hydroxyl group. Sulfur is going to connect to our carbon chain, and sulfur is connected to a hydrogen. They are different electronegativities, and so that sulfur is going to carry a partial negative. That hydrogen is going to carry a partial positive. So again, a sulfhydryl group will add polarity to a molecule, but it'll remain non-ionic. One thing that you're going to learn when we start talking about proteins is that, in addition, sulfhydryl groups have this unique ability. If you have two sulfhydryl groups, okay, on two different molecules, they can be involved in a redox reaction such that a covalent bond forms between the two sulfur groups and we lose the two hydrogens. This is called a disulfide bridge. It's a covalent bond that links those two sulfurs together, and this will be an important reaction for the structure of many proteins. And we'll look at that a little bit more when we talk about proteins. Next, phosphate groups. This is a big, fat, chunky group. We have a phosphate atom connected to 1, 2, 3, 4 oxygens. It's this oxygen here that's then connected back to that carbon. And take a look at this. This is 2 ions. That oxygen is negatively charged. That's a full-blown ion. That ion there is a negatively charged ion, full-blown ion. And so when you connect this big functional group with 2 giant negative charges onto a hydrocarbon, you very much change the chemical properties of that molecule. And this is going to be extremely important, a functional group, when we start discussing biochemistry and energy within the cell. Okay, and when we start talking about ATP, that P in ATP, adenosine triphosphate, is this right here. So this guy we are going to come back to quite a bit when we get into chapters 8 and 9 and 10. So it is polar and it is ionic. Okay, now we come to the 2 most important functional groups, the ones that I want you to practice the most with. They are the carboxyl group and the amino group. So the carboxyl group first is different than a carbonyl. So don't get confused between a carboxyl and a carbonyl. Carboxyl group is a carbon that is double bonded to an oxygen and then single bonded to an oxygen and a hydrogen. This is more than just a carbonyl with a hydroxyl group, right? This is one of those examples of an emergent property. When you put a double bonded oxygen and a single bonded OH onto the same carbon, you've got a series of emergent properties that aren't around with just a carbonyl and just a hydroxyl group. So this is a great example of emergent properties. And now the special thing about a carboxyl group is that when you put it into a aqueous solution of physiological pH, it reaches an equilibrium. Some of it stays like this in its protonated form. That hydrogen is on that oxygen, and some of it breaks down. Some of it deprotonates. So now you've lost that proton, right? The hydrogen's electron stays here. That's why we have an extra negative charge here now. But the hydrogen's neutron and the hydrogen's proton, the positively charged aspect of that hydrogen, have come off. So what we've done is, in this equilibrium here, we have some that's deprotonated and we have some that's protonated. This should very much remind you of what we learned regarding buffers and weak acids. Carboxyl groups are weak acids, and carboxyl groups can be buffers. Now, I want to go through something here. When we look at something like a carboxyl group, so let's go ahead and write that again. So here's our carboxyl group. There's our C double bonded to an O and then to an OH. And then when we put that into an aqueous solution, it reaches an equilibrium such that we have our carbon double bonded to oxygen and then our other oxygen minus, because we lost that proton, and then there is our proton. Okay, there are a whole bunch of terms that we can apply to this equilibrium, this reaction. The first pair, ionic and non-ionic. Okay, so which one is ionic? Right, it's the one right here. So this is ionic. And this is therefore non-ionic. Let's do protonated, deprotonated next. Okay, so which one of these sides is the protonated state of the molecule and which one of these is the deprotonated state? So the proton, when we talk about protonated, deprotonated, the proton, we're talking about that hydrogen that can be attached and become detached. Well, it's this hydrogen here, which means in this particular case, it's detached, so this is the deprotonated state. Which means here must be the protonated state. Okay? And finally, we have another couple terms that we can apply here. The acidic form and the basic form. Now let me define these in a very specific way for you because it can be very confusing if you don't define it properly. The acidic form of something is the form at which it can donate a proton. Not that it has donated a proton, but it can donate a proton. It's really important that you remember that definition. So which one of these two can donate a proton? This one here, right? It's still bound to its proton. That means it can donate it. So this is the acidic form. And then the basic form is the one, we define it as the form that can accept a proton. Not the form that has already accepted the proton, but one that can accept. So that is this one right here. So we say this is the basic form. The basic form is the one that can accept a proton. The acidic form is the one that can donate a proton. So make sure that you can apply these three pairs of terms to a carboxyl group. Okay, let's go back and take a look at amino groups. Now, amino groups are just as important as carboxyl groups, meaning they're very important. And amino groups are the nitrogen, which is attached to a carbon, bound to either 2 or 3 hydrogens. Okay, so what we can have is a situation where here's our amino group that is deprotonated. It's only bound to 2 hydrogens. And then here it is protonated. It's bound to that 3rd hydrogen. The difference here, though, is that it only takes its ionized form when it is protonated, when it is bound to the 3rd hydrogen. That's when it takes on a positive charge. So let's do the same thing that we did for the carboxyl group for an amino group. So I'm going to come over here, and then I'm going to erase all this. Let's go ahead and write NH2 plus an H plus. Whoops, that plus is in the wrong place. Yield, and this is equilibrium, this is reversible, NH3 plus, and that's it. All right. So now we have this equilibrium. When you put in something with an amino group into an aqueous solution, some of that amino group will soak up some of the protons to become this, and some of them will stay deprotonated. So again, let's take a look at this equilibrium, and now let's apply these pairs of terms. Ionic. Which one, let's call this A or B, is in its ionic form? An ion means a full charge. There's our full charge, so this is ionic. Non-ionic. All right, let's look at our basic form, acidic form. Remember, let's start with the acidic form. The acidic form is the form that can donate, that hasn't donated it already. It can donate a proton. Well, if we look in this direction here, NH3 plus going to NH2 plus an H plus, that's where we donated the proton. So this then is the acidic form, and this, the basic form, is the form that can grab onto a proton. So this NH2 can grab onto that proton to become that. So this is the basic form. And finally, protonated, deprotonated. Hopefully that's pretty obvious by now. The protonated is in regards to an amino group. It's talking about that 3rd hydrogen, not those first 2, but the 3rd one. So here we've got 3 hydrogens, so this is protonated. And this only has 2, and there's that 3rd, it came off, so this is the deprotonated state. Protonated. Good. Make sure that you can do this, that you can write out this equilibrium of an amino group, you can write out this equilibrium of a carboxyl group, and that you can assign these 3 pairs of terms to each side of the equilibrium. All right, let us finish off with the last one. And that is, this is the most different of all of the ones. This is a methyl group, and what we're doing is we're essentially adding another CH3 unit to that hydrocarbon. And CH3, carbons and hydrogens, are equal electronegativity. So this functional group, in contrast to the other ones, is nonpolar. So when you add this to a molecule, you aren't giving it any polar effects. You're sort of just changing the shape of that molecule. That is going to play quite a big role in the expression of genes and something called gene silencing that we will talk about so much later in the course. Okay, so again, make sure that you can go through this video once again, and that by the time you've digested this material, you can explain the properties of carbon that make it so important for biology, that you identify the different hydrocarbon isomers, right? The branched form, the chain form, the ring form, and so on and so forth. And that you can identify the functional groups on molecules and describe what chemical properties they contribute to the molecule. And then that includes those three pairs of terms: ionic, non-ionic, basic form, acidic form, protonated, deprotonated. And from that, you will have a much better understanding of the 4 macromolecules that we are about to go into: proteins, lipids, carbohydrates, and nucleic acids.